So we're covering meiosis today. Let's review from last time. Mitosis. Mitosis, mitosis, mitosis. This is just replicate the DNA and copy. It's a photocopy mechanism. This is for the entire cell to make an exact replica. We use this to grow, repair, and it's functionally called asexual reproduction. I make a really big point about it being asexual reproduction because meiosis, by comparison, is for sexual reproduction. That's combining genes. The cell cycle, cells spend most of their time in what phase? Interphase. So interphase is like 80-90% of the cell's life. Unless it's a cancer cell, then it's likely sprinting through this phase. The piece of the cell that is the least amount of time is mitosis, kind of a set amount of time. I don't know how long it takes, and there's probably some variation, but it doesn't take ages to go through the mitotic phases. What are the mitotic phases?
Interphase, anaphase.
Almost. Come on, how do we remember in order?
Interphase.
IP on the mat. So interphase is before mitotic phases. So technically the mitotic phases are P, mat. Prophase, metaphase, anaphase, telophase, and then finally, for a brief second, cytokinesis before it launches back into G1. So we're covering sexual reproduction. That's for meiosis. It makes genetically unique gametes. Gametes are also called gross, yucky germ cells. So these are our germ cells. They're haploid in number. They have half the genetic material because you can only chip in half to the offspring. So they can't be a photocopy of you, that's a clone. Each offspring is half of each parent. All right, from just the review, meiosis produces blank number of daughter cells. A, B, C, or D? I need you to weigh in.
Oh wait, I don't know how to tell.
You got this? It's 4 haploid cells. So remember that we just go through mitosis, but we do it one more time. So it creates 4 cells, but each of them are half the genetic material.
I still don't understand haploid and diploid that well. I know it's like 1 and 2. I don't get
It has to do with the number of amount of genetic material. So you are a combo of mom and dad.
it. Yes.
Mom chipped in a haploid cell, an egg. Dad chipped in a haploid cell, a sperm. They fuse together to make you. You're basically mitosis of this again and again and again. So you're diploid. Genes from mom and from dad.
But then when
You make an egg.
combine inside, then they just produce haploid cells, right?
During which process? In your ovaries, they're making haploid eggs.
No, like in my arm.
In your arm, they're photocopying an exact copy of a diploid cell, so they're making other diploid cells.
I don't understand at all.
Let me see if I can go to the next thing, and it might help.
Okay.
So, our key root words. The big ones that I care that you know are haploid. Haploid meaning single. This is half the genetic material. We use these in germ cells. Diploid meaning double. This is, the word ploid means form. So this is how many chromosomes. Remember how it was all identified based on what they looked like in a microscope? So we're like, this one has 1 and this one has 2 in terms of its form of chromosomes. Yeah.
So when the sperm cell and the egg cell, they're both haploids, when they combine, does it become a diploid?
So when 2 haploid cells come together, they fuse and form a diploid cell. So because 2N is just 2 N's,
Okay.
so it's N plus N. When they fuse together a sperm and an egg, that's a diploid cell. They both brought in an N, so they become 2N.
And then it's diploid from then on out.
You got it. Until we get to ovaries and testes, who are like, ooh, we're going to mix it up a little bit.
Yeah.
Okay, meiosis means to lessen the stages. So stages of lessening is what it really means. There's not less stages, there's way more stages than mitosis. So what in the stages is lessening? The amount of genetic information. We start with a a diploid cell. 2N. And then we double it. So for like a brief moment, we sort of have 4N. It's not called 4N because the chromosomes they're redundant of each other. Does that make sense? So it's not technically 4N, but you can think of it as 4N for a second. So, and then they divide. And you have 2N cells. Sounds a lot like mitosis. But then we divide those one more time through the exact same process. The second time we divide them, it is exactly like mitosis. The first time we divide them is weird. We throw in a couple of ways to mix it up. There are ways to increase genetic variation. But the end result is half the genetic material in each cell. So that's where it comes from to lesson. Okay, a muscle cell of a typical organism has 32 chromosomes. How many chromosomes will be in a gamete of that same organism? So the first thing I want you to pay attention to is, they said muscle cell. Does that make any freaking difference? It just means body cell. They could have said somatic cell, but they're trying to trip you up with just saying that it's one type of somatic cell. Doesn't matter. It's somatic no matter what. So it's telling you how many are in the diploid condition, 32. It's asking how many are going to be in the haploid condition. If diploid is 2N, what's N? 16. So you get it. It's the one time that our Blue Book question comes up twice. So, as always, you can get a shot of the screen if it's easier to get a picture of. So, what other word could you use for sex cell?
Germ.
Germ cell. What other what other name could you use for body cell? Somatic. So those are totally okay words to use because they're the exact same thing. Growth. Which one of these cells is important for growth? Go for it.
The mitosis.
Mitosis. This is where you just double the cells. When you take baby, like loaf of bread size Zannie, and you make 6-foot Zannie, there's a lot of mitosis that happened. So here are methods of mixi-matchy. We have independent assortment. There's a slide for each of these. I'm just previewing it. What do you know when your teacher puts a color-coded list on the board? 1, she loves colors. 2, it's important and you have a test early next week. So no pressure. Know these. Draw a star next to them. These are your key ways of increasing genetic variation. First one, independent assortment. This happens in metaphase 1. So what I want to show you is that interphase, totally the same.
Yeah.
Prophase, at least as far as this goes, totally the same. It turns out this is where crossing over happens. But where they line up and how they're going to end up splitting into the eventual cells of the 4 daughter cells is important. So lining up on the metaphase plate is really important. It creates that random chance of how they line up is another way to sneak in variety. Cedar.
Is interphase the exact same as mitosis?
Yes,
So G1 as G2? Okay.
nailed it. Interphase is the same throughout.
Okay.
Can you see that they've already crossed over? So when must that have happened?
And
Prophase. So these are the 2 really key steps for genetic variation. In our first round of divisions, in meiosis, where we create the 2 daughter cells that later split becoming the 4, after interphase, prophase 1 and metaphase 1 are a big deal for genetic variety. So we have, just by virtue of how they line up, whether it's the blue ones on one side and the red ones on the other, we have 2 separate options. Now imagine that with 23, or 46 ultimately, because this is the first round. So you see that the variety, just in showing the chromosomes here, creates a lot of variety. Okay, here's your cheat sheet, and this is what's posted online. What I think is really helpful about this one is I color-coded. The key step here is crossing over. This happens in prophase 1. Is that like a really easy multiple choice question? So it's just like, where does this happen? Know it. Metaphase 1, this is where you get your independent assortment. It has to happen when they're lined up on the metaphase plate to be yanked apart.
Sorry, real quick. You said this was online,
all of this is. I added our sneak cheats. So M for metaphase is when they meet in the middle. That's our independent assortment. A for apart. This is for the full chromosomes being ripped apart. This is a change. Remember in mitosis, we're ripping the sister chromatids in half. Here we're ripping full chromosomes apart. The X together rather than the two halves, we're separating the full X because we need that for the next step. You see here how we start with still kind of a diploid form? We need to then split that into two and then two haploid cells. This is exactly like mitosis. The first round of meiosis is where we sneak in the variety. The part of meiosis that is most similar to mitosis is A, B, C, or D. What is D?
I think it's like a.
I know, it's a really fun word, but we've never used it. So can we rule out D?
Yes.
Okay, so now we have a 33% shot? Okay. Is it anaphase one?
Oh.
No, that one's weird. So we're between A and B. Thank you, A and C. I was just trying to figure out how not to flip you off while I do this. So, which one is the most similar? Where do we get the weirdo meiosis things? Is it in 1 or 2?
2. I mean, sorry, one.
You were answering the question rather than the one I just asked. So yes, our weirdo meiosis genetic variation occurs all in meiosis 1, meaning that meiosis 2 is our very most like mitosis. But I walked through the steps of how I think of it because I think that so many people go, I know the answer, and they don't read the question. Start by crossing out the ones that you know for sure are like, no, that doesn't make any sense. And really think about not just the answer to the question, but what I've provided. Because in a multiple choice question, it's not what's the right answer, it is which is the most correct.